Isso. Aynen. Aynen. Aynen. Aynen. İşte. Hiç kimse yapamam. Non mi ricordo.
Farmers all around. generation time. It takes typically 5 months or so to go from seed to seed. And that's not a very good thing if you're a PhD student working on wheat. It's not like Drosophila or yeast, for that sake, where you can do lots of genetics very fast because of the short generation time. So, confronted with that problem, Lee Hickey, a researcher in the University of Queensland, he came up with a process called speed breeding. And we worked with Lee Hickey to perfect this process. The plant on the left is about 5.5 weeks old. It's grown under our standard conditions in the John Innes Center, where I used to work. This plant here on the right is also 5.5 weeks old, and it's grown under speed breeding conditions. And under these conditions, we can go from seed to seed in just 8 weeks, 2 months. So what is the trick? We have done away with the old sodium vapor lamps that produce a lot of light in the green and the yellow part of the spectrum that you can see up in the right-hand corner. And that is not very useful for photosynthesis. Photosynthesis works better in the blue and the red part of the spectrum. So we replaced the old sodium vapor lamps with LED lights optimized for blue and red light. And then we increased the light intensity, and we kept the lights on for 22 hours to trick the plants into getting very little sleep. And under these conditions, we were able to cycle the plants very fast in just 8 weeks from seed to seed. And it works not just for wheat, but also for barley and many other crops and their model derivatives. The other challenge of working with wheat is that the genome is very large. It's about 150 times or so bigger than the Arabidopsis thaliana genome, the lab rat of the plant world. And there's a lot of genes in wheat compared to Arabidopsis. And within these 120,000 genes, we have the immune receptors, the genes encoding disease resistance. Now, it turns out that most disease resistance genes that have been defined by genetics and subsequently cloned in plants over the last 30 years, they encode a type of immune receptor known as a nucleotide binding site leucine-rich repeat. And armed with that knowledge, you can compare these genes with each other. You can compare the sequences, and we call them NLRs for short. So you can compare NLRs, and there are certain motifs that are represented again and again. And these motifs are numbered up here. And so if you take those motifs and scan a genome, you can very quickly annotate, you can find the NLRs in the genome. And there's about 3,400 NLRs in the wheat genome. So the genome is using a lot of its potential to encode for these genes. It really cares about immune receptors. Now, traditionally, genes have been cloned in wheat using map-based cloning, so using genetics. This is, it's pretty slow, and you rely on recombination to get close to your gene. And so some years after starting this journey, we introduced mutational genomics into wheat. And what we did was we would take a wheat strain where we had defined by genetics that there was a single disease resistance gene against a certain isolate of the pathogen that we were working with. And so we would take a big bag of this seed and mutate it with a chemical mutagen, typically EMS, grow up the mutant population, infect it with the pathogen, and then let the pathogen do the work to identify susceptible mutants. And then we would use enrichment sequencing. We would use exome capture to to sequence just that small proportion of the genome encoding the NLRs. And this works very well. We could design RNA probes that are complementary to all the NLRs and then fish out sequences corresponding to these NLRs and then use next generation sequencing or Illumina sequencing to sequence the NLR-ohm. And this worked very well. We got about a 1,000-fold enrichment. The other way that we could reduce the complexity of the wheat genome was to collaborate with Jaroslav Dolocel in the Czech Republic. At the time, he had fine-tuned the art of chromosome flow sorting in wheat so that he could get pure preps of single chromosomes. In the diploid, that would reduce the complexity 7-fold. In hexaploid wheat, that would reduce the complexity 21-fold. And so what we would do, we would sequence the resistant parent, align all the 3,400 candidate immune receptors, and then we would sequence a handful of mutants. And we would line them all up, and we would look for all the NLRs that have mutations in them. And we would, in particular, look for an NLR which would be mutated in all of the independently derived mutants. And the probability of that occurring by chance alone is very low. So that would be our candidate disease resistance gene. And in fact, that worked very well. There's some publications on the right where we reduced this concept to practice. And the one at the bottom I like to point to, where we cloned YR5 and YR7, that was work done by a PhD student in the lab, Clémence Marchal, and she managed to clone 2 disease resistance genes within the confines of her PhD, which at the time was quite a remarkable feat. Now, some of you will notice that some of the genes we cloned were for stem rust. Stem rust is a devastating disease of wheat, which can cause epidemics and destroy the crop. And historically, it's been associated with crop failure and famine. And for many years, scientists and farmers have realized that stem rust has an alternate host, which is the barberry bush. And it requires the barberry bush to go through sexual reproduction. And so for many years, farmers and communities around the world have tried to destroy the barberry bush. This is a campaign poster from the early 20th century in the US, where they were encouraging farmers and communities to destroy the barberry bush. And they would do this by pouring salt onto the plant or going out and using horses to yank the plants out of the soil. Here's a more sinister campaign poster, which came in the wake of the Bolshevik Revolution, where the barberry or the stem rust was likened to the red-handed anarchist of the grain fields. Going further back in history, ancient farmers in Greece and in Rome, they had a god called Robigus, and they would sacrifice a red-haired animal every spring to pacify Robigus and protect their crops. And this is our first paper that we published on cloning stem rust resistance genes in wheat. And as you can see, the Robigalia Festival was held on the 25th of April, and we published our paper on the 25th of April. Now, don't tell me you don't believe in Robigus now. Some communities in Italy, they still practice this. I'm sure you can think of a fiery red-haired politician to replace instead of the poor dog, but we'll leave it at that thought experiment. Don't go any further. Okay, so we tried to see if we could adapt this method of mutational genomics to the wild relatives of wheat. And here we have an experiment that's ongoing to generate a mutant population of a wild grass called Aegilops sharonensis. You would starve to death if you depended on sharonensis for your daily bread, but it has remarkably good disease resistance. So we started this project. We had to do a lot of so-called breeding in sharonensis to genetically isolate a single stem rust resistance gene. Then we had to bulk up the population. And then we had to harvest several thousand seed, 20,000 seed, and we had to peel them. It's very difficult. It's so difficult. It's almost like putting bamboo splinters under your nails, a horrible process. Then we mutated the seed, and about 6,000 of the seed survived the mutagenesis, and they grew very well in this glasshouse. We took up the whole glasshouse in the John Innes Center, but not a single one of the plants were fertile. None of them set seed. That was very disappointing. And in fact, it's very difficult to mutate a diploid because there's no genetic redundancy in the housekeeping genes. This is a lot easier in hexaploid wheat, where you have 2 or 3 copies of all the housekeeping genes. You can hit them really hard with mutagenesis. So we had to give up this experiment, and we realized that we would need different approach. We would need to do genetics or create populations that would not require laboratory controlled genetic structuring. And so we decided that we would go and use the hand that Mother Nature had already given to us. And that's represented up here in the left-hand corner. This is the genetic structure of another wild wheat, the one I introduced in the earlier slide. This is Aegilops tauschii, the D genome progenitor. And here we can see the natural genetic diversity that has been generated over hundreds of thousands of years by natural recombination in the field and natural spontaneous mutagenesis. And so we reckoned that if we would phenotype all the individuals in this genetic structure with a single isolate of the pathogen, we would get a range of phenotypes. And then we reckoned that if we used our resistance gene enrichment sequencing, we could sequence the NLR-OME or complement of all of these individuals. And then we would be able to do association genetics to derive correlations between phenotype and genotype, and hopefully identify candidate disease resistance genes. So to reduce that concept to practice, we decided to work on this genetic structure, and we did resistance gene enrichment sequencing on 150 accessions of Aegilops tauschii. And then we did association genetics for disease resistance. Our first trial was with a isolate or a race of the pathogen called RKQQC. This was a positive control because this isolate of the pathogen is diagnostic for a disease resistance gene called SR33, which had already been cloned after introgressing into wheat. So this was very useful because we could test our method. And lo and behold, we got a single peak in our genome-wide association plot, which corresponded to SR33. So this was very exciting. But of course, you can, when you're doing bioinformatics, it's easy sometimes to find what you know you're looking for. So we had to do it with the without positive controls. And so here's another one with a different isolate of the pathogen called TPMKC. And here we were able to identify SR45, which is actually another positive control, but we were also able to identify a peak which genetically corresponded to another gene that hadn't been cloned called SR46. And we were later able to confirm that this candidate gene was indeed SR46. And finally, with QTHJC, we were able to identify a candidate gene for SRTA 1662, and we were later able to confirm that this was indeed the right gene. So in conclusion, with a bit of sequencing and phenotyping and association genetics, so bioinformatics, we were able to identify all the 4 major stem rust resistance genes that had been genetically defined in this species over the last 40 years. Okay, so this was all very well. This allowed us to clone disease resistance genes encoding NLRs. But we really wanted to be able to clone genes that were not encoded by NLRs and be completely unbiased in our approach. So therefore, we decided we had to sequence the whole genome of every member in the panel. And this was something I proposed quite some years ago now. And back then, it was considered a very large task to sequence so many individuals with such a big genome. And so we had to form an international consortium of crazy people like myself working on these wild relatives. And with this consortium, we were able to sequence not just the original panel, but enlarge it to 242 accessions. And with that, we were able to do association genetics for phenotypes other than disease resistance. And so here we've looked at variation in trichome density on the leaves. We've looked at spikelet number, and we've also looked at a pest wheat curl mite, and we looked at a classical disease, powdery mildew. And interestingly, for powdery mildew, we got, as you can see, a very discrete peak on chromosome 7. And under this peak, there were no NLRs, but there was a wheat tandem kinase. And we've later confirmed that this wheat tandem kinase is indeed the disease resistance that confers this resistance to powdery mildew. So that was very gratifying because we really demonstrated that now we could clone disease resistance genes that are not NLRs. And that was published a few years ago in Nature Biotechnology, and it was a very large consortium effort. Now, recently I moved from the UK to KAUST in Saudi Arabia. And with generous funding from KAUST and also with support from the Open Wild Wheat Consortium, we expanded the size of the panel to nearly 500 accessions. And we also generated some very high quality assemblies using long read PacBio sequencing for 46 of these accessions. And then now we are working with the community to look at lots of different traits that are of strategic interest. And we continued our interest in disease resistance. And with these new resources, we were able to clone a leaf rust resistance gene called LR39, which also encodes a wheat tandem kinase. And we were able to finally give SRTA 1662 a gene designation, SR66. And that was published just last year in Nature. And again, another large consortium effort. And so now it's like being a little child again and going into the sweet shop. We've cloned a lot of genes. And what do we now do with all those disease resistance genes? We're spoilt for choice. So we have collaborated with Mick Aliff and his postdoc Ming Lu in the CSIRO in Australia. And they have taken some of the genes for stem rust that were cloned by us or other members of the community, and they have generated a stack called the Big Five. And as you can see on Mick Aliff, you've got to be really strong and big to make these stacks. And this was transformed into a cultivar of wheat fielder, which is very susceptible to stem rust. And then it was tested in the field in Rosemont, in Minnesota, in the States, in the US, by Brian Stephenson and his postdoc, Oadi Matni. And as you can see, the non-GM is completely susceptible to stem rust in the field, whereas the Big Five is completely immune. And Brian, who has worked for more than 30 years on stem rust, he said he had never seen anything like it. So this was a very, very gratifying and exciting moment. Now, there is a bit of an elephant in the room, and that is that this is a GM product. And there's a lot of socio-political opposition to GM wheat. And in an opinion piece that I published a few years ago with Kanwa Palduga, we likened GM wheat to a GM orphan crop because a lot of our major crops like cotton has BT, maize has BT, and soy is also GM, canola is GM, but wheat is a GM orphan crop. But I would say there is light at the end of the tunnel. There's a small biotech company, largely unheard of, in Argentina, and they've been working on the quiet for nearly 20 years to develop a GM product in wheat. They have introduced a gene from sunflower which confers drought tolerance. It increases the yield in low input environments by about 16%. And recently, they managed to deregulate the so-called HB4 wheat. So this is very exciting. And lots of jurisdictions, they are now accepting HB4 wheat. And importantly, in 2021, HB4 wheat was accepted by the authorities in Brazil. And that was a really important watershed moment in GM wheat because Argentina exports a lot of wheat to Brazil, and that makes this GM crop commercially viable. And now jurisdictions around the world are jumping on the bandwagon and they are deregulating HB4 wheat. And I would argue once we have a GM crop, it will be easier. It will lower the barrier to introduce additional GM traits, such as for disease resistance. And so we are working with Bioceres in Argentina, and they do care about, this is a picture of HB4 wheat on the right and the control on the left, grown under drought-prone conditions. And as you can see, the HB4 wheat grows very well. But it does suffer from stem rust. So we're now trying to introduce a GM stack for stem rust into the HB4 background. But it is not so straightforward politically. A lot of these genes that have been cloned have been protected by patents. And you can see there are two main players. There's the CSIRO in Australia. They've cloned a lot of genes and they have protected these with patents. And for many years, I was funded by the Two Blades Foundation. And they have also patented most of the genes that I cloned. So that complicates your freedom to operate. And you can see that many of these genes, patents, they will still last for very many years. But the landscape is rather checkered, whereas jurisdictions like Australia, the USA, and the European arena, they have the patents, they cover these genes. In South America, you can use most of these genes without any restriction. So that opens a possibility to introduce this technology into South America without being encumbered by patents. And so now to some predictions for the future. I think over the next 5 years or so, we're going to see increased activity in gene cloning in wheat. I predict that we will clone another 200 disease resistance genes for rusts alone. I think we will identify the corresponding effectors that these immune receptors recognize in the pathogen population. This will allow us to study the efficacy of all the cloned genes very carefully. And that is important because when we understand the efficacy, we can design judiciously stacks that are going to be more durable. And I think it won't be long before we can then generate a triple rust stack, so that we would create a wheat that would not have stem rust, stripe rust, or leaf rust. And I think this will likely happen as part of a public-private partnership, but it is a complicated exercise which will require a lot of diplomacy and a lot of politics. And so one of my final conclusions is that when you work on wheat, there's a little bit of science, maybe about 20% science and 80% politics. And so finally, I'd like to thank some of the many funders who have supported me over the years. I could still need more funding. So one day I'm going to steal the handbag from an old lady crossing the road, an old rich lady. That's what I was thinking about anyway. But instead, I went to Saudi Arabia, and I'm now using money from a dead man, a very visionary individual, King Abdullah, who sadly passed away some years ago. He was very visionary. He funded or founded the King Abdullah University of Science and Technology. It's a very well-funded institution with researchers from all over the world. There's about 100 different nationalities in KAUST. And it's really a lovely place to do science. And this is the view from my office, by the way. No, I'm joking. This is my group. I'd like to thank my new group in KAUST. And that's it. Thank you very much for your attention.
Thanks so much, Brande, for the entertaining, wonderful, exciting talk. And I expect that there will be a lot of questions here. And who want to start? Or are there any? Yeah.
Thank you very much, Brandy. Eclopes tavşan is how it calls? Ajda adı mıydı? Ancestor of bread wheat, yes? So if we do GWAS in Eclopes tavşan, it's work in bread wheat or not? Sometimes.
So what is the question? We can do genome-wide association studies in KAUST.
Yes.
We can identify candidate genes. And then the question is, do those genes work in wheat?
Yeah.
We can incorporate the genes into wheat by wide crossing, and we can make synthetic hexaploids or direct crosses with Taoxiai and introduce the genes. So you can do it in a natural way that is exempt from any regulation. And I think most of the time the genes, they do work in the hexaploid background. Sometimes you find that they don't work. And that might be due to dilution when you put it into the hexaploid background. And so maybe you then need to find a way to overexpress that gene or screen many individuals to find one where that expression works. The percentage, I don't know. We test most of our genes by transgenesis, and most of the time that works. But we do find occasions where it doesn't work as well in the hexaploid background. And I will, in my next presentation, I'll give an example. I'll get close to an example of that. Some resistance genes are digenic, and they require two factors. They require a sensor factor that can perceive the presence of the pathogen, and they require a helper factor that can signal downstream resistance. And if they're not genetically linked and you just introduce one of them, you may leave behind the other one, and then the gene doesn't work. So sometimes you need two genes.
Thank you. Good presentation.
Are there any questions?
Hi, Brenda. Great science and a nice delivery as usual. Hopefully you have cloned many genes genetically, and we hope that these great wealth of genetic data will be transferred into practical breeding soon. So the main question is, my question is now, so you have cloned many genes, including the gene for curl mite. So do you have a co-segregating markers for simple mass system?
Yeah, so we haven't formally cloned the gene for curl mite yet. We have used GWAS to define a genetic region which by definition should contain the gene. And this is not our area, someone else is following up on that. Generating markers we have for some of the genes we've cloned, like the YR genes, the YR5, YR7. As part of that publication, we generated genetic markers. We've also generated genetic markers for some of the blast resistance genes that we cloned in collaboration with Paul Nicholson. But it should be straightforward to take the sequences of the genes that we've cloned and use those to generate perfect markers. And yeah, the sequences are published. Anyone can do that. Thank you.
Thank you very much, Dr. Brande. So my question, we know a lot of resistant genes cloned and identified, and some of them not cloned yet. Did you focus to clone some newly identified genes nowadays?
Yes, so some of the genes that we've discovered in the wild relatives are so-called new. They haven't been genetically defined and designated. That's one of the advantages of working on some of the more wacky stuff like Agelops sharonensis. I have a love-hate relationship with sharonensis. It is so difficult to grow. The agronomy is so poor. It has all the hallmarks of being undomesticated. It has extraordinary genetic diversity for disease resistance, and very little of this genetic diversity has been introduced into the wheat breeding pool. And so we hypothesize that if we focus on some of these wild relatives that haven't been used in breeding, we may discover new broad spectrum disease resistance genes that have not been, let's say, exploited by the breeders and overcome through deploying them as singletons and selecting for variants in the pathogen population that are virulent.
Okay, so from the questions from biotechnology, recently we had a colloquium in our university. It was a talk about NLR engineering, and what do you think about the future of NLR engineering? Would it be possible to do really engineering of NLRs for resistance?
Yes, there are several emerging examples of being able to engineer NLRs to widen the spectrum of recognition or indeed recognize altogether new effectors. And for that to be successful, you really need the 3D structure of the NLR and the effector and any other signaling components that work in concert with the NLR. So I think the future is bright. And if I were Demrust now, I would be shaking in my spore.
Great talk. So coming from the effector side, so do you think that the effectors are engaging directly with your kinases, or is it an indirect effect? I mean, do you have any examples for how that works?
Yeah, in fact, I have a slide on that in my next talk. But in some cases, the interaction is direct between the receptor and the effector. And in many cases, it's indirect. And I think one of the most seminal contributions in this field was made in 1998 by a postdoc in the group of Jonathan Jones, Eric van der Biesen, who hypothesized that in many cases, the interaction is indirect. And that in fact, the effector is targeting a pathogenicity target in the host, and the NLR is guarding that pathogenicity target and can detect the effect of the effector on the pathogenicity target. And so it's an indirect interaction. And this is a beautiful way of explaining how a host can defend itself against a myriad of effectors with just a few thousand, or in many cases, just a few hundred receptors. Because there's a finite number of cellular hubs that are targeted by effectors. Jochen.
Brande, thank you very much. It's a really great talk. And in particular, I like your visionary style. And I have two questions. Second questions will be somehow visionary. You talked about these receptors, but when you showed the survey of cloned genes, you also touched on, without commenting on, durable resistances, LR34, LR67. Wouldn't it be a good strategy to stack them in these panels of resistance genes?
Yes, the so-called APRs, adult plant
durable
yes.
resistances, which rely on entirely different mechanisms like transporters.
Yes, completely. In fact, one of the genes in this big five is one of these broad spectrum resistance genes. It was a little hidden. Because we used in that slide, I used a different designation. I think it's LR34, which is also known as SR55 or so, because it confers triple rust resistance, but that is in the stack. And now the latest thing is that Mick Aliff he's got his big five. He's made a new stack called, I think it's Fearless 5. And if you combine Big 5 and Fearless 5, you have 10 stem rust resistance genes. I think this is going to be a very tough nut to crack for the pathogen.
Absolutely, yeah. This in part answers my second question. So the breeders community is a quite nice community, and they think a lot together, but when it comes to individual success, they are egoistic. And if a breeder has a successful cultivar with just one resistance, which belongs to your stack, then this may be overcome, and another breeder has a second, and so on. Yeah. And of course, even though you would have 5, there is a risk that these will be overcome, but of course 10 is another league.
Absolutely. That's you put your finger on it. I think we need to clone more disease resistance genes from these wild, wispy, insignificant looking grasses that the breeders, they wouldn't even touch. There's a lot of genetic diversity out there. And the technology for cloning these disease resistance genes is improving. We started off driving a Ford Fiesta 15 years ago. We're now driving a Ferrari. And I think we're about to step into a spaceship. And so I really believe that in 5 years or so, we'll have another 200 resistance genes cloned, including their corresponding effectors. And then we can study on a worldwide scale the efficacy of these genes. And I think we'd be able to design some stacks that are going to be so difficult for the pathogen to overcome. I'm not saying they will ever be overcome, but we can have more stacks in the background, next generation stacks.
Maybe I have general question. So is there any pathogens or disease specific to triticum? If yes, and do you think they have also kind of friendly conversation, and then how do you recognize the host plant, and then how like host plant also can support the pathogen to establish on the plant?
So if there's any pathogen of wheat which is specific only to wheat, well, it takes something like powdery mildew. There's a wheat adapted version that grows more or less specifically on wheat and durum wheat and some of the closely related grasses. So they tend to be quite specialized and adapted, these pathogens. I think maybe turn the question round and ask, is there any grass that doesn't support rust? And in fact there is, and that's rice. The rice is very interesting because there's no rust that grows on rice. And so people have wondered for many, many, many years, what is the nature of this non-host resistance? And so it's difficult to do genetics on non-host resistance. But, you know, maybe now with CRISPR, we saw some wonderful presentations yesterday on CRISPR. And so could we use that technology to remove all the NLRs from rice and see if it would now be susceptible to rust? I think that would be very interesting, and that would allow us to do genetics on rice for non-host resistance and understand what's the basis of that. And maybe learn from that, you know, how could we turn wheat into a non-host?
Thank you. And maybe thanks so much, Brande.
Thank you.
Lovely discussion. Now we invite our next speaker, Jochen Kumlin. He will be talking about double coupler technology, which is really interesting.